Our today's topic is nutrition and growth of embryos. In embryophytes, the young plant contained in the seed begins as a developing egg cell formed after fertilization and becomes a plant embryo. So, embryo is an unborn or unhatched new organism in its early stages of development. Plant embryos grow and receive nutrition in a number of ways, including from endosperm, suspensor, gametophyte, or through photosynthesis, germination, cotyledons, dormancy, or nutrient balance, etc. Plant embryogenesis begins with an asymmetric cell division, resulting in a smaller apical cell and a larger basal cell. Most of the plant embryos develop from the apical cell. The suspensor develops from the basal cell. This suspensor anchors the embryo to the endosperm and serves as the nutrient conduit for the developing embryo. Further cell division leads to the globular stage. The three basic tissue systems, dermal, ground, and vascular, can be recognized at this point based on characteristic cell division patterns. I can see here the apical basal pattern means organization of organs along the apical basal axis. And then radial pattern, here organization of the three basic tissue systems, dermal cells, then ground tissues, and vascular tissues. The globular shape of the embryo is then lost as the cotyledons begin to form. Just look at this diagram. Here, 2 cell stage of the embryo, right? Then 2 to 4 cell stage. This is globular stage of the embryo. So this globular stage is then of the embryo is then lost as the cotyledons begin to form. The formation of 2 cotyledons, I can see here, 2 cotyledons in dicot gives the embryo a heart-shaped appearance. This is early heart shape and this is proper heart-shaped appearance. So upright cotyledons can give the embryo a torpedo stage. You can see here in this figure, this is torpedo stage. So upright cotyledons can give the embryo torpedo shape, and by this point the suspensor is degenerating, you can see here, and the shoot apical meristem here and root apical meristem at this point are established. These meristems will give rise to the adult structure of the plant upon germination. Further growth of the cotyledons results in the torpedo shape, band stage, and then at last mature embryo stage. At this point, embryogenesis is arrested and the mature seed desiccates and remains dormant until germination. During their early growth, the embryos of all vascular plants exist as virtual parasites, depending for nutrition on either the gametophyte or upon an initially triploid tissue, the endosperm. Early nutrition of the sporophyte in ferns, horsetails, club mosses, such as Lycopodium, is clearly provided by the gametophyte. In these groups, the young sporophyte produces a multicellular structure, the foot, which remains embedded in the tissues of the gametophyte throughout early development, withdrawing nutrients. In Slaginella, the pteridophytes, the gametophytes is sexually distinct. The female gametophyte develops within the wall of the megaspore and never escape completely. After fertilization, the zygote cleaves and the outer cell produces a long suspensor that pushes the embryo deeply into the tissues of the gametophyte. A foot is then formed and further development of the embryo continues at the expense of reserves transferred to the megaspore from the sporophyte generation. In the pines, the female gametophyte develops within the tissues of the nucellus and acquired abundant food reserves. The pro-embryo forms after a period of free nuclear division in the zygote, and the ties of cells above the basal 4 then elongates to form a suspensor. The suspensor pushes the embryo deep into the gametophyte. During embryogenesis, the gametophyte continues to grow and to accumulate food materials, which is transferred to the embryo or remain as reserves in the seed. In angiosperms, the embryo is nutritionally dependent upon the endosperm. The reserves accumulated in the endosperm include carbohydrates, lipids, and proteins. As reserves accumulate, the nuclei of the endosperm cells may undergo deformation and degeneration. In many plants, the growing embryo consumes the endosperm before seed maturation. In others, the tissue persists in the seed, providing a reserve for the developing seedling after germination. In cereals, starch and proteins are catabolized upon the initiation of germination by hydrolytic enzymes secreted from the alluring layer. Next is, how do embryos get nutrition? First, we will discuss the role of gametophyte. In early development, vascular plant embryos rely on the gametophyte for nutrition. Plant embryos receive nutrition from gametophyte in the number of ways. First, by producing a foot. The embryo can produce a foot that withdraws nutrient from the gametophyte. Then transferring reserves, the gametophyte can transfer reserves to the embryo. Then providing unnutritive tissue. The female gametophyte can provide a nutritive tissue that feeds the embryo. For example, in gymnosperms, the female gametophyte is large starch-filled tissues that surround the embryo. Next is role of endosperm. In angiosperms, a double fertilization event initiates the development of two distinct structures, the embryo and the endosperm. The endosperm plays an important role in supporting embryonic growth by supplying nutrients, protecting the embryo, and controlling embryo growth by acting as a mechanical barrier during seed development and germination. Its structures and function in mature dry seed is divergent and specialized. Among different plant species, the endosperm is a tissue that surrounds and nourishes a plant embryo in the seeds of flowering plant or angiosperms. The endosperm provides nutrient to the embryo in the form of starch, oils, and proteins. Cells of the endosperm are triploid and arise from the fusion of the polar nuclei with sperm nucleus through a double fertilization process. The endosperm is rich in carbohydrates but also contains proteins and lipids. It is a critical part of seed and seedling survival as it helps establish dormancy and control, longevity, and physical resistance. Next is how the endosperm provides nutrition to the embryo. The embryo secretes signal to the endosperm to induce the degradation of the seed reserve and to promote endosperm weakening during germination. The endosperm is capable of sensing environmental signals and can produce and secrete signals to regulate the growth of the embryo. Thus, germination is a systemic response that involves bidirectional interactions among the embryo and endosperm. In seeds like peas and beans, the endosperm is absorbed completely by maturity. The embryo is then nourished by the fleshy cotyledons as it germinates. In seeds like wheat and castor beans, some of the endosperm remain until germination. The cotyledons are thin and membranous here and absorb food stored in the endosperm when the seed germinates. The liquid endosperm, in the case of coconuts, it contains important growth substances. A subset of endospermic tissues are composed of living cells even after seed maturation and play an active role in the regulation of seed germination. Now, growing embryo mainly receive nutrition from endosperm and gametophyte, but there are some other ways that provide nutrition to the growing embryo, like process of germination, cotyledons, suspensor, photosynthesis, and nutrient balance, etc. So first of all, germination. The embryo germinates and begin to grow when conditions are favorable, such as when it senses enough water, oxygen, and other minerals. Then cotyledons. As the embryonic plant develops, cotyledons absorb the food supply and continue to nourish the plant. If the nutrition is stored as endosperm, the cotyledons are generally small and undeveloped, whereas when the cotyledons are enlarged, there is little endosperm in the mature seed. Then dormancy. Seed dormancy is defined as uh, an intrinsic block to germination of seed under otherwise optimal conditions. So, dormant seeds maintain an embryonic cell identity and desiccation tolerance. The termination of dormancy and induction of the germination program are both cellular phases, means transition from the embryonic to vegetative state and the discrete induction of plant growth. Now, what is embryonic cell identity? It's a complex gene regulation program that allows embryonic stem cells to remain pluripotent and self-renew. Means these embryonic stem cells are unique in their ability to differentiate into almost any cell type in the body and to divide into cells with the same developmental state as the parent. Then nutrient balance. A proper amount of each nutrient is important for plant health. A deficiency or overdose of nutrients can result in plant death. Next is suspensor. Suspensor is a filamentous or spherical organ that orients the embryo's absorptive surface toward its food source. It may also serve as a nutrient conduit for the embryo. Last is photosynthesis. Mature embryos are fully autotrophic, means they can photosynthesize and synthesize all the organic molecules they need to grow.